He is also director of the Center for Infection and Immunity, an academic laboratory for microbe hunting in acute and chronic diseases. Lipkin is internationally recognized for his work with West Nile virus, SARS and COVID-19. Walter Ian Lipkin is the John Snow Professor of Epidemiology at the Mailman School of Public Health at Columbia University and a professor of neurology and pathology at the College of Physicians and Surgeons at Columbia University. Hen became a clinical clerk at the UCL Institute of Neurology in Queen Square, London, on a fellowship, and an intern in medicine at University of Pittsburgh. He completed a residency in medicine at University of Washington, and completed a residency in neurology at University of California, San Francisco. He conducted postdoctoral research in microbiology and neuroscience at the Scripps Research Institute from 1984 to 1990, under the mentorship of Michael Oldstone and Floyd Bloom. In his 6 years at Scripps, Lipkin became a senior research associate upon completing his postdoctoral work, and was president of the Scripps Society of Fellows in 1987. Lipkin was born in Chicago, Illinois, where he attended the University of Chicago Laboratory School and was president of the Student Board in 1969. He relocated to New York and earned his BA from Sarah Lawrence College in 1974. At Sarah Lawrence, he felt that if I went straight into cultural anthropology after college, I'd be a parasite. I'd go someplace, take information about myths and ritual, and have nothing to offer. So I decided to become a medical anthropologist and try to bring back traditional medicines. Suddenly I found myself in medical school. Returning to his hometown Chicago, Lipkin earned his MD from Rush Medical College. In 1978, he borating with researchers at Sun Yat-sen University in China. Dr. Lipkin had also advised the Chinese government and the World Health Organization during the 2002-2004 SARS outbreak. Dr. Lipkin described his own infection with the SARS-CoV-2 virus, beginning mid-March 2020, which resulted in a case of COVID-19 and necessitated his recovering from the illness at home. On the podcast This Week in Virology, Lipkin has earned the reputation of a master virus hunter due to his speed and innovative methods of identifying new viruses, and has been lauded by National Institute of Allergy and Infectious Diseases Director Dr. Anthony S. Fauci as director of the Center for Infection and Immunity at the Mailman School of Public Health. Lipkin, from the onset of the COVID-19 pandemic, has led CII researchers' Ent tenure at Columbia as the founding director of the Jerome L. and Don Green Infectious Disease Laboratory from 2002 to 2007, which transitioned to the John Snow Professorship he holds at present. Lipkin was the Louise Turner Arnold Chair in the Neurosciences at the University of California, Irvine from 1990 to 2001 and was recruited shortly thereafter by Columbia University. He began his a major step towards identifying and studying new viral pathogens that emerge locally throughout the globe. A major node in a global network of investigators working to address the challenges of pathogen surveillance and discovery. Dr. Lipkin has trained over 30 internationally based scientists in these state-of-the-art diagnostic techniques. A physician-scientist, Lipkin is internationally recognized for his work with West Nile virus and SARS, as well as advancing pathogen discovery techniques by developing a staged strategy using techniques pioneered in his lab. These molecular biological methods, including mass tag PCR, the green chip diagnostic, and high-throughput sequencing, AR. His research focused on pathogen discovery, using unexplained hemorrhagic fever, febrile illness, encephalitis, and meningoencephalitis as targets. He is the principal investigator of the autism birth cohort, a unique international program that investigates the epidemiology and basis of neurodevelopmental disorders through analyses of a prospective birth cohort of 100,000 children and their parents. The ABC is examining gene-environment timing interactions, biomarkers and the trajectory of normal development and disease. Lipkin also directs the World Health Organization Collaborating Center for Diagnostics in Zoonotic and Emerging Infectious Diseases, the only academic center, and one of two in the U.S., that participates in outbreak investigation for the WHO. Lipkin is the director for the Center for Research in Diagnostics and Discovery, under the NIH Centers of Excellence for Translational Research Program. The CRDD brings together leading investigators in microbial and human genetics, engineering, microbial ecology and public health to develop insights into mechanisms of disease and methods for detecting infectious agents, characterizing microflora and identifying biomarkers that can be used to guide clinical management. Lipkin was previously the director of the Northeast Biodefense Center, the regional center of excellence in biodefense and emerging infectious diseases, which comprised 28 private and public academic and public health institutions in New York, New Jersey and Connecticut. Within this consortium, Lipkin was co-chair of CDC Steering Committee of the National Biosurveillance Advisory Subcommittee. The NBAS was established in response to Homeland Security Presidential Directive 21. Public Health and Medical Preparedness. Thai Cooperative Research Center for Emerging Infectious Disease. The Guangzhou Institute for Biomedicine and Health. The Eco Health Alliance. Tetragenetics. And 454 Life Sciences Corporation. He is honorary director of the Beijing Infectious Disease Center, chair of the Scientific Advisory Board of the Institut Pasteur de Shanghai, and serves on boards of the Australian Biosecurity. Lipkin served as a science consultant for the film Contagion. The film has been praised for its scientific accuracy, where he poses a potential correlation between eosinopenia and bacteremia in diagnostic evaluations for a bacteremic patient. While at UCL, he worked with John Newsom Davis, who was utilizing plasmapheresis to better understand myasthenia gravis, a neuromuscular disease. While not quite a medical anthropologist, Lipkin specializes in infectious diseases and their neurological impact. His first professional publication came in 1979 during the time of his fellowship in London as a letter to the editor at the Archives of Internal Medicine. The approach of looking for a virus genes instead of looking for antibodies in infected people, as a way to speed up the diagnosis process. By the mid-1980s, Lipkin had published 2 papers specifically about AIDS research and transitioned into utilizing a more pathological approach to virus identification. He identified AIDS-associated immunological abnormalities and inflammatory neuropathy, which he showed could be treated with plasmapheresis, and demonstrated early life exposure to viral infections affects neurotransmitter function. In 1981, Lipkin began his neurology residency and worked in a local San Francisco clinic, which was about the time AIDS began to affect the local city population. Because of the social view of homosexual people at the time, very few clinicians would see patients with these symptoms. He was watching many patients fall ill with AIDS. It took years for scientists to discover the virus responsible for the disease. I saw all of this, and I said, we have to find new and better ways to do this. It was during this epidemic that Lipkin 2 infections could selectively alter behavior and steady-state brain levels of neurotransmitter mRNAs. The next step was to look for infectious agents which could be used as probes to map anatomic and functional domains in the central nervous system. In 1989, Lipkin was the first to identify a microbe using purely molecular tools. During his time as chair at UC Irvine, Lipkin published several papers throughout the decade dissecting and interpreting Bornavirus. Once it was apparent the viral controlled study of the link between BDV and psychiatric illness was completed by the researchers at Columbia University's Center for Infection and Immunity in a joint effort that concluded there is no association between the two. Lipkin noted that it was concern over the potential role of BDV in mental illness and the inability to identify it using classical techniques that led us to develop molecular methods for pathogen discovery. Ultimately, these new techniques enabled us to refute a role for BDV in human disease. But the fact remains that we gained strategies for the discovery of hundreds of other pathogens that have important implications for medicine, agriculture, and environmental health. By the mid-1990s, it was asserted that Borna disease is a neurotropic negative-strand RNA virus that infects a wide range of vertebrate hosts, causing an immune-mediated syndrome resulting in disturbances in movement and behavior. This led to several groups across the globe working to determine if there was a link between Borna disease virus or a related agent and human neuropsychiatric disease. The group was formally called Microbiology and Immunology of Neuropsychiatric Disorders, and the multi-center, multinational group focused on using standardized methods for clinical diagnosis and blinded laboratory assessment of BDV infection. After nearly 2 decades of inquiry, the first blinded case RTS nearby the initial reported cases were also the initial points of entry into the United States. During the 5 years after the first reported case, Lipkin worked on a study with the National Institutes of Health and the Wadsworth Center at the New York State Department of Health to determine how a vaccine could be developed. While they had some success with the immunization of mice with PRMELPs, as of 2018, there is still no human vaccine for WNV. In 1999, West Nile virus was reported in 2 patients in Flushing Hospital Medical Center in Queens, New York. Lipkin led the team identifying West Nile virus in brain tissue of encephalitis victims in New York State. It was determined potential routes for the spread of West Nile virus throughout New York originated from predominantly mosquitoes, but also possible from infected birds or human beings. There is a high likelihood the 2 international airpo IDEMIC identify the gaps in science and develop a strategy for containing the virus and reducing morbidity and mortality. This brought the development of real-time polymerase chain reaction technology, which essentially allowed for the detection of infection at earlier time points as the process, in this instance, targets the N gene sequence and amplify the analysis in a closed system. This markedly reduces the risk of contamination during processing. Test kits were developed with this PCR-based assay analysis, and 10,000 were hand-delivered to Beijing during the height of the outbreak by Lipkin, whereupon he trained local clinical microbiologists on the proper usage. He became ill upon his return to the U.S. and was quarantined. Chinese scientists first discovered the severe acute respiratory syndrome coronavirus in February 2003. But due to initial misinterpretation of the data, the information of the correct agent associated with SARS was suppressed in the outbreak. Investigation had a delayed start. Advanced hospital facilities were at the greatest risk as they were most susceptible to virus transmission. So it was the classical gumshoe epidemiology of contact tracing and isolation that brought swift action against the epidemic. Lipkin was requested to assist with the investigation by Chen Zhou, vice president of the Chinese Academy of Sciences, and Xu Guanhua, minister of the Ministry of Science and Technology in China, to assess the state of the EP.